Molecular adaptations to marine environments. Marine organisms inhabit some of the most extreme and variable environments on Earth, from the crushing depths of the ocean to frigid waters of the poles and the toxic surroundings of the hydrothermal vents. To survive and thrive in these unique conditions, they have evolved a variety of molecular adaptations. These adaptations occur at multiple biological levels, including protein structure, enzymatic functions, genetic regulation, and cellular stress responses. This report explores key molecular strategies that enable marine life to maintain homeostasis, ensure functionality, and adapt over evolutionary time. Now that we've covered the introduction, let's move to the first key topic, which is protein structure and function under extreme conditions. Protein structure and function under extreme conditions. Proteins are essential macromolecules that must maintain their structure and function under environmental extremes such as high pressure, extreme temperatures, and chemically hostile habitats. Deep sea organisms. Proteins in deep sea species are adapted to withstand hydrostatic pressures exceeding 1000 atm. These proteins exhibit more rigid and compact structures, often with increased hydrogen bonding and hydrophobic interaction to prevent denaturation. Hydrothermal vent species. These organisms encounter both high pressure and extreme temperatures, often exceeding 100 degrees Celsius. Their proteins have evolved higher thermal stability through amino acid substitutions that enhance intramolecular interactions. Polar organisms. In cold environments, proteins tend to have increased flexibility to compensate for reduced molecular motion. This is achieved by decreasing proline content and increasing surface charge to prevent freezing and maintain catalytic efficiency. Now, let's move to the second key topic. Enzymatic adaptations. Enzymes in marine organisms must function effectively despite drastic changes in temperature, pH, salinity, and pressure. Thermophilic enzymes. In hydrothermal vent organisms, enzymes possess a more robust structure with increased ionic interactions and disulfide bridges, enhancing thermostability. Psychrophilic enzymes. In polar organisms, enzymes are more flexible, with lower activation energies and altered active site dynamics to maintain catalytic activity to near freezing temperatures. Barophilic enzymes. These deep sea enzymes function optimally under high pressure, often showing altered volume changes during reactions and enhanced pressure tolerance in their tertiary structures. Genetic and genomic adaptations. Marine organisms exhibit a range of genetic and genomic modifications that underpin physiological resilience and environmental specialization. Gene duplication. This process allows for redundancy and innovation in protein functions, particularly in stress response and metabolic pathways. Horizontal gene transfer, or SGT. Especially in microbial communities, HGT introduces novel genes for detoxification, metal resistance, and metabolic adaptations. Gene regulation. Marine organisms often display dynamic regulatory systems, including inducible expressions of stress-responsive genes and environmental sensors that trigger protective mechanisms. Stress response mechanisms. Marine environments impose a variety of stressors, including pollution, UV radiations, temperature shifts, and low oxygen levels. Heat shock proteins, or HSPs. These molecular chaperones prevent protein aggregation and assist in refolding damaged proteins during thermal or oxidative stress. Antioxidant enzymes. Enzymes like superoxide dismutase or SOD, catalase, and glutathione peroxidase neutralize reactive oxygen species generate under hypoxic or polluted conditions. Detoxification systems. Cytochrome P450 enzymes and multi-drug resistance proteins help metabolize and expel harmful substances, contributing to pollutant tolerance. Biomineralization. Biomineralization is the process by which marine organisms form calcium carbonate shells, silica skeletons, and other mineralized structures. Examples: molecular control, genetic regulation, ecological importance. Molecular control. This involves specialized proteins like matrix proteins and chitin synthase that template mineral deposition, regulate crystal growth, and control polymorph selection. Genetic regulation. Genes responsible for ion transport, organic matrix production, and pH regulation are tightly controlled to ensure precise mineral structure formation. Ecological importance. Biomineralization plays a critical role in species protection, buoyancy regulation, and reef building, and is sensitive to changes in ocean chemistry such as acidification. Conclusion. Marine organisms demonstrate an extraordinary capacity for molecular innovation in response to environmental challenges. Understanding these adaptations not only provides insight into evolutionary biology and ecological resilience, but also holds potential for biotechnological applications. From deep-sea extremophiles to polar survivors, the molecular strategies employed by marine life exemplify the complex interplay between biology and the environment. And that's all for my report. Thank you for listening.